would fit the horizontal.
We'll see.
All right. I guess we can get started. Good afternoon, everyone. Thank you for joining us. It is my distinct pleasure to introduce today's seminar speaker, Dr. Yang Liu, who's visiting us from University of Wisconsin-Madison. Because he's an old colleague of mine, I can just call him Yang. So Yang did his PhD from University of Southern California under Dr. Feng Yu Sun before starting his postdoc at Bill Noble's lab at University of Washington. I am very fortunate that while I was being a postdoc at Bill's lab, Yang and we overlapped, and I remember having very interesting lunches with him where we would talk about frustrations of work, but also all sorts of interesting ideas. In fact, I do owe Yang for introducing me to optimal transport because he went to NeurIPS, came back and said, "Oh, Ritamra, I think this paper might be of interest to your project." And that basically started a whole research direction in the lab. So thank you, Yang, for that. So Yang's research is really interesting because he combines a lot of interesting computational methods with very thorough and robust statistical modeling and comes at all sorts of interesting challenges in the biomedical domain. So with that, and then I forgot to mention that after his postdoc, he first went to University of Waterloo as an assistant professor and then decided to move back to the US and is now at Wisconsin-Madison. So with that, thank you, Yang, for joining us. And let's give a round of applause.
Thank you so much for your kind introduction and invitation. I'm very, very excited to be here. This is Brum is an amazing place. So I met a lot of old friend, new friend, and legendary researchers. So let's start. Okay, so today I will introduce you some recent works we have accomplished in my lab. So this will be 3 stories about how to find needle from haystack. So let's start. So a couple of years ago, the whole world has been greatly impacted by COVID-19 pandemic. So at a very early stage of pandemic, where we did not have this antigen test. So for the healthcare system and patient, a reliable and rapid test for the COVID-19 patient will be desirable. Okay. So for this purpose, researchers all over the world has spent tremendous effort to detect different biomarkers for diagnosis purpose. And these biomarkers, span all our human body. As a computational biologist, one natural question immediately arises, that is, can we use AI to accelerate such very tedious biomarker detection process? Although this idea seems very promising, but in reality, this is very, very challenging. As a study point out that surprisingly, there were hundreds of so-called AI diagnostic tools on the market. Surprisingly, none of them was clinically useful due to various flaws. Okay, so as I point out by another study that basically these AI tools, they try to do diagnosis based on some kind of shortcut, such as what are the water markers of the sample, what are the batch information from samples, instead of relying on really plausible biological signals. So without such a flawed model, biomarker detection as a much harder task will just fail. Okay, so this is very disappointing. Although the challenges we faced, I will still argue that AI and machine learning is revolutionizing current biomedical research. Conventionally, people follow so-called hypothesis-driven paradigms. That is, biologists will just guess and hypothesis, say this could be a candidate biomarker for COVID-19 diagnosis, and then they will try to collect the data to do some experiment, and then to see if what they observe align with their hypothesis. Mostly not. So they need to evaluate, calibrate, and their hypothesis iteratively until find a reasonable solution. This is really, really tedious and slow. Nowadays, I would argue that we are in a right time in the sense that we have more and more data with final resolution. We try to complement this traditional hypothesis-driven paradigm by so-called data-driven paradigm. That is, this AI expert will try to build a very powerful machine learning model from this large volume of data which has been collected historically. Okay, this model can be very smart and it can in principle do a lot of prediction and evaluation in silico, so that even though it might not directly hit to the right answer, but not too off. So therefore, the calibration and evaluation will be much, much efficient. So therefore, it will be much effective. Okay. To make it more concrete, to facilitate such kind of research paradigm, on one hand, we still need to follow the so-called prediction axis. That will be the input will be all omics data as our measurement in our human body, and we also have we call it genotype, and then we also have something we are interested, some trait or phenotype, right? The disease status. We want to build a AI model to predict the phenotype from the genotype by capturing the subtle patterns from this data, which sounds straightforward, but as I shown previously, this is still non-trivial. But what my research more interested is in the other axis. That is, say we have a pretty good AI model for this genotype to phenotype mapping. What can we do next? So we want to interrogate this machine learning model to generate a bunch of disease-specific hypotheses which can be tested experimentally. So this hypothesis could be in the form of, what are the disease-specific biomarkers? Oh, sorry. What are disease-specific biomarker interactions or causal relationship or even higher order pathways? Of course, this is very challenging. So the challenges mainly lies in the following aspect. First of all, we are dealing with highly parameterized black box machine learning models. So without knowing why and how it make a prediction, right, it's very difficult for biologists or clinicians to believe it. And secondly, if we want to use this model to generate hypotheses, it could be overwhelming. For example, if we have 10,000 genes, the combination, pairwise combination, will be 10,000 choose 2. That will be 50 million, let alone go even higher order. So it's explosive. And meanwhile, among these huge, huge haystack of this hypothesis, the real ones are really, really tiny. The majority are false. Okay, how to prioritize this important hypothesis, and meanwhile, how to make sure not getting too many false positives. This is highly, highly non-trivial. And lastly, we are dealing with heterogeneous data, ranging from different omics, right? And each of these data, they have different customized processing. Ideally, we want to piece them together to make these hypotheses mutually support each other. That will be make it more reliable. So in short, these challenges lies in the aspect of modeling, hypothesis, and data. So today's talk will tackle each of these one by one. Okay. So in the next maybe 30, 40 minutes, I will go through 3 stories we have accomplished in the past few years. And these 3 stories will span several most important aspects of modern biomedical research. So the first story will be how to build models to discover hypotheses interpretably. Okay. And then the second story will be how to prioritize this hypothesis statistically rigorous. And lastly, how to deal with this omics data reliably. So I hope that by the end of this talk, I can convince you that it is indeed possible that we can directly enable data-driven biology by directly get rigorous hypothesis from data. Okay, so let's start from our first story. So the first story will be related to the single cell. Okay, everyone, I believe, work very much on single cell. Basically, single cell, they can profile, they have a lot of applications. They can profile in cell resolution what happens in your human body, both spatially and temporally. So this is a typical analysis pipeline for single cell RNA-seq data. So from this individual, we get a bunch of a population of cells containing a different cell type, mixture of cell types. We sequence them, and what we end up with is this large cell by gene matrix. Although this matrix is very high dimensional, but the cells actually they reside in a low dimensional space we call manifold. Here is no manifold more than elsewhere. So essentially here, each of these point, the cell clouds, they are just like colored differently, indicate, different cell types. And usually, biologists are interested in answering the following question, like, what are the genes like distinguish different cell types? So in order to answer this question, usually the pipeline, the analytical pipeline works as follows. The input is this cell by gene matrix, and we try to use some kind of dimensional reduction technique such as UMAP, t-SNE, or even deep autoencoder, try to project and denoising the cells into this low dimensional space. And within that space, we can see that, there will be, the structure will be more obvious, that we hope that each of these cluster will correspond to different cell types. And then we just, try to do some kind of statistical analysis to measure for each of these genes, right, how much it differentiates from one cell type to others. Okay. So this conventional pipeline has some kind of caveats. One is, when we try to do this kind of dimensional reduction to the latent space, right? So how reliable is the UMAP, t-SNE, or different dimensional reduction technique? How many uncertainty may it cause? And secondly, if we want to assign, let's say, one cell to cell type A or B, it's easy for something in the middle, something inside of the cluster, but it's very hard, for something in the middle, right? How much it will affect the outcome. And lastly, when we try to analyze, for example, each gene, how specific it is to certain cell type, they just treat individually, right? And then maybe do a correction afterwards. But, their dependency definitely matters, but mostly ignored. So in order to address these kind of caveats, if we look at, all the existing methods, of course, they're definitely not all of them, but several representative ones. Essentially, they suffer more or less of these caveats. So therefore, a couple of years ago, we proposed a method called ACE. So essentially, it's trying to achieve the benefit of both. Okay. So from high level, of course, I will explain details in the next few slides. But high level, the idea is it try to join modeling all the separate steps along this workflow, very long bioinformatics workflow, such as dimension reduction, clustering, and differential analysis. So here's the idea. So the input is always, a cell by gene expression matrix. And of course, usually we can use some data-driven way to find out what will be the best cluster number. Or, usually we just use this Louvain, right? We don't really need cluster numbers. And so essentially the first step is we want to do the dimensional reduction. Here, we not only want to project this data into a low-dimensional space, but also want to do some other things like imputation of the missingness, try to do the denoising, right? So essentially, this is something a very sophisticated deep autoencoder model can do. Okay, of course, this is meant to be model agnostic, but we just use one proposed by the Smitas group at Yale. So the idea is, similar to UMAP, t-SNE, or p-SNE to do the dimensional reduction, but meanwhile to do other things. Okay. Once we have this project all the cells into a low dimensional space, and then we want to do the clustering. For example, here we can use, say, for simplicity, this k-means clustering algorithm. And in order to join explain both, the uncertainty in k-means as well as the uncertainty in dimension reduction, we use a technique, this technique called the clustering neuralization. The high level idea is we try, because remember what is k-means clustering does, right? Essentially, it's try to minimize an object by trying to make with the cells within a cluster denser, and then pushing the cells from different cluster apart. So essentially, it will try to minimize a object function. So essentially, we will try to rewrite this object function in a equivalent neural network. So therefore, here, each of these block will be, the soft assignment of k-means clustering in this neural logic. And then the output will be how likely, for example, one this particular cells be assigned in k-means clustering, to this cluster assignment. Why we want to do this way? Because in this way, we can directly concatenate the dimension reduction as well as this clustering in the same neural network framework. And with that, we can possibly do a lot of things. Okay. For example, we want to say, okay, and then we want to say, what will be the difference if we want to move one cell from one cluster to the other cluster, say from blue to uh, to yellow, right? And then we want to directly propagate. We want to find some kind of what will be some kind of the changes we made to their gene expression so that it will make the cluster assignment to change. So essentially, it's like if I want to move the cell from here to here, what kind of change to make in the gene space? Why is that important? So I will give you an analogy. This analogy is in the computer vision domain. For example, we have this image 8, okay? And then, of course, a machine learning model will classify this as 8. And then we ask a question, no, I don't want this to be 8. I want this to be 3, okay? What kind of changes I need to make in the pixel space so that makes this 8 look like 3, okay? So essentially what we need to do is, okay, we just try to make this two loop disconnected by subtracting this two small area. Okay, and then this is what we end up with. Although this is still look like 8, but essentially this is 3. So I would argue that these small changes I call perturbation will encode the key characteristic difference between these two labels. Similarly, we want to use this intuition. If we want to move from one cluster to the other cluster, and what will be the characteristic changes in the gene to make that happen? And we argue that will be the signal for the marker genes. Okay. So this is what we do. Essentially, we will just ask question, if we want to move from, this blue cluster to this red cluster, this is the change we want to make in their cluster assignment. And then how do we propagate these changes all the way back to the concatenate models, through both this is the cluster model and this is the dimensional reduction model. And what we end up with, by all these propagations, we end up with is this perturbation. Okay. And this perturbation will measure how likely and how relevant each gene to this particular change. And then this is, what we do, we essentially we get this perturbation. We use this L1 norm to make sure this is minimized. And meanwhile, we try to rank it. Oops, something's broken here. So we try to rank it so that we hope that higher ranked genes will be more relevant to a specific cluster of interest. Now let's do some sanity check. Okay, we still start from something more straightforward. Let's still do the computer vision task. Let's say, we want to change from 1 to 7, right? What kind of changing we need to make? Essentially, we just, intuition tells us, we just need to add this top bar, right, to the 1. So it's essentially to make it look like 7. And of course, this change is inherently symmetric. For example, what if I want to change from 7 to 1? Essentially, it suggests that, okay, we need to remove this top bar. Okay, so this is quite straightforward. Of course, we can do this for a lot of, the digit pairs to make sure that, yeah, this is realistically and intuitively makes sense. And now we want to apply to some simulated biological data set. So we use a toolkit called SimSim. So the idea is we generate this 20 causal genes to form this kind of structure. Okay, so it is worth mentioning that this causal gene is not biological causal, but just informative enough to form these 5 cluster structures. So we want to test the capability of our method. Essentially, which will also add like 100 totally non-informative noise genes, as well as we just take this random weighted sum with the combination of 20 causal genes to have 100 dependent genes. Essentially, they're just redundant, but information with different weighted combination. Okay, so and then overall, when we sum all these 220 genes together, they still form this well-structured 5 cluster structure. The task here is, we want to rank the cluster-specific genes by relevance, and then we want to measure, right, how well these top-ranked genes can capture this underlying cluster structure. So we use this matrix called the mean Jacob distance. The basic idea is we want to capture if we have a subset of top-ranked gene, how well it can preserve the nearest neighbor of all the cells if we use all the genes. So the higher matrix means, this cluster is less, the nearest neighbor, because this is distance. So essentially, the neighborhood information is less preserved. The lower means, even if you use a subset of the genes, still the cell neighborhood semantics are preserved. And then, so here's the result. So this is the baseline. If we use the initial 20 causal genes, this will be the mean Jacob distance is preserved. Okay, and this is the result of ACE. You can see when we use 20 genes, it's even lower. That means it can find even a better combination of these dependent genes, even more informative than this initial 20 causal genes, a better combination. Okay, and compared to other 6 methodologically different methods, each of which will use a ranking, we can achieve the best in terms of preserving this local semantics. And lastly, we apply our method to a real melanoma data set. So the idea is, there are 2 cell types, just benign versus malignant. And of course, the label is determined not by the cluster, otherwise there will be double dipping. So it's determined by some orthogonal information, the copy number variations. And so this is you can see there are way more genes than the number of cells. Usually in math, this is called the high dimensionality problem. This is quite challenging. And then still our task is try to rank what will be the malignancy specific genes, right? So we have these 2 matrix. We want to see 2 matrix. One is how well these top ranked genes can discriminate between malignant versus benign. On the other hand, we don't want too many redundancy. We want this gene we get out is quite diverse. So for the first matrix, we want this, still to be very discriminative for this subset between these 2 different types. So the idea is for this top rank gene we retrain an SVM model with RBF kernel to see how well it will separate 2 cases. And then this high discriminativeness will be reduced to the predicted accuracy. This can be measured by area and the ROC curve. Because they are and then the other metric is I want to make it diverse. So essentially, we just, for all the genes, we look at their correlation of their expressions, and then we want their correlation to be smaller the better. So essentially, we use this average Pearson correlation. If the select genes, they are less correlated across all the cells, that means they are more diverse. And we can see that as a result, our method can achieve almost perfect discriminative power and with almost the lowest redundancy. So essentially, they will be the best trade-off. Okay, so to summarize this part, okay, so the goal is, ACE itself is try to identify a subset of signals, to as a panel to characterize different single cell type. Okay. But what I want to emphasize is that, although we are analyzing a seemingly separate and disjoint analytical pipeline, but we can use this technique called neuralization, try to make them into a unified framework. What's the benefit of that? Essentially, it can jointly consider, all the uncertainties together and attribute to the interpretation. So, and then from high level, what we achieved is, we demonstrate, we can indeed interpreting a bioinformatics workflow and to generate hypotheses. The idea is if we have data, we can, either do direct modeling or we can use this model neuralization to make it into a deep neural network, and then to use that to generate hypotheses. Of course, still a long way to go from hypothesis to scientific discovery, right? How to make sure that, this hypothesis, for example, here is a ranking list of, say, 10,000 genes or even more. How to make sure that, we identify the most important and reliable ones. And this leads us to the second story. Okay. So the second story is about, provide confidence estimation for the hypothesis. So of course we can do like for individual features like we had in ACE, but we want to do something more complicated. Okay, so in the second story, we care about how to deal with these so-called non-additive interactions. So these non-additive interactions, by definition, is just like, these interactions which cannot be decomposed into smaller units. For example, in these toy equations, like variable x1, x2, or x3, x4, they cannot be further decomposed into smaller units. But for this, the x5, x6, although they are in this kind of the product, but actually they can be decomposable in the log space. So essentially, they are not non-additive. What we only care about non-additivity, because in reality, when we deal with gene-gene interaction, drug-drug interaction, and so on, we care about this kind of synergistic effect, right? What is the extra gain if we add two things together? In addition, compared to we treat them separately. So this is the setting. Okay, usually we have still a sample by feature matrix. In the previous sample, this could be a cell by gene matrix. It's a bit generic. And then we have some phenotype of interest. Okay? So let's assume we already have a pretty okay machine learning models, but the goal here is never about a more accurate predictive model. It's more about once we have this model, what do we do next? We want to interrogate this model to compare each pair of the features. Okay, as our hypothesis, we induce a ranking list with hope that higher ranked interactions will be more explainable to the disease of interest. Okay, remember this ranking list is huge. Okay, say we have 10,000 genes. This ranking list could be 10,000 choose 2, 50 million. So for biologists, they have their own budget. They cannot try everything. Okay, maybe they can try 10. Okay, so now the problem is, how do we choose the 10 interaction as hypothesis, which is most confident with minimum error? Okay, this is the setting. The idea is we try to provide a confidence estimation for each of these hypotheses in the ranking list. This metric we call false discovery rate, or FDR, tells you to what extent each of these hypotheses could be wrong. Let's say if we choose the top 3, that means, at most 1% of chance it can be wrong in a data-driven way. How do we do that? So essentially, let me introduce this metric called false discovery as a more used jargon or concept in stats community. So the idea is, it's the ratio between, the denominator is the totally accepted interactions as our hypothesis or discovery, and the numerator will be the false positive. Okay, in the setting of, let's say we have a ranking list, and then we assume the higher the better, and we need to cut off some way, right? Let's say we just cut off somewhere, and then all the pairs above this cutoff threshold will be our accepted interactions, shaded in green. Of course, we are not God. We don't know among these interactions how many of them are wrong. We don't know. Okay, we need to estimate. So traditional ways is we need to estimate through two-stage procedure. One is we need to somehow calculate the p-values of these interactions by some kind of permutation test, and then we need to do a multiple hypothesis correction, say using Benjamin Hirschberg, right, to estimate the FDR. But remember, we are dealing with arbitrary, highly parameterized deep neural network. So it's highly non-trivial to get a null distribution to handle p-values. So making this problem extremely hard. Okay. So what did we do? Okay. So essentially, what we do is the first step is once we have this model, we use some kind of off-the-shelf so-called explainable AI method, try to measure, for example, for each of these pair of features, how much they as a pair contribute to the prediction of disease status. Okay? Or we can say for each individual features, how much itself contribute to disease prediction. We can use a lot of off-the-shelf method to do that. For example, we can calculate the first order, second order derivative, as a measurement. Or we can use, for example, more fancy way like Shapley values. Yeah.
I just have a clarification question. Does the pairwise importance comes from just calculating the correlation?
Not correlation. So essentially, it's like like hashing. For example, what will be the partial derivative of this function, this this function with respect to feature I and j. Yeah. So essentially just calculate the second order hashing. You can do that. So this is something off the shelf. But one thing and take away is this off the shelf method cannot directly be used. So this is a very simple illustration why. Okay, say we have a pair of features, each of interest, say I and j, together with other, say let's say if we have test sound feature, plus this will be test sound minus 2 other features. They jointly will go through this predict model to predict the disease status. So what so-called feature importance measure do is this essentially try to project the total contribution of all test thousand features back to each single feature axis. For example, if we project everything onto this feature I axis, that will be so-called the marginal importance of feature I. If we project everything onto this feature J axis, there will be so-called the individual importance of feature J. And similarly, if we project everything onto this IJ plane, this will be feature I and J as a whole, how much they contribute to the final prediction. But what we really care about is not about this red arrow. It's not about this blue arrow. What we really care about is something, extra. This, in this green arrow. This green arrow basically tells you what's the extra thing. You add these two things together minus the jointly. So what's the extra effect, extra gain? They act separately versus jointly. So this is something we call the synergy. Okay. So based on the type of these green arrows, we can characterize, whether this is a synergistic effect or whether this could be a repressive effect, something like that. And following this intuition, we just try to get, how to measure this non-additivity, this, the effect encoded by these green arrows. What we do is we start from, still what we have is the observed, what will be the pairwise importance by projecting to the plane, and what will be the individual importance by projecting to the single axis. And then we try, this is something we want. This is the non-additivity effect. This will be, we just try to subtract from this observed pairwise importance. And by, they are called the label dependent marginal effect. Of course, because they are comparing apple to oranges. So we need some kind of data-driven scaling we learn from data to make sure they are really comparable. And meanwhile, we found that there are some extra bias. For example, if a feature is just simply has a larger magnitude, it will cause the importance to be higher. But actually, this is not really more important. But instead, it's just have higher magnitude. Essentially, we want to also subtract this kind of label independent feature bias. And as a result, I just get some details how we optimize that. But as a result, what we're leaving is guaranteed to be the non-additivity. Okay? Once we have that as the ranking matrix, now the next question is how do we control the FDR? We use a concept called the knockoff. The basic idea is this is the input, right? It's assembled by features. The basic idea is for each of these column, in other words, a feature, we want to construct some kind of realistic null. Okay? So essentially, for each of these column here, we want to construct a realistic null as control. The basic idea is they are realistic, and they can serve as a control to estimate the false positive in our discovery. And then, of course, this is manually constructed. They are definitely wrong, right? But what do I mean by realistic? Okay, so in details, so essentially these null features, they are constructed in a very smart way. They perfectly mimic the dependency structure of the original features. For example, we want these fake features to capture this intra-correlation structure of the input. And meanwhile, we want these features to capture this inter-correlation structure of between themselves and the input. So therefore, of course, in other words, it just capture the first order summary statistics, second order, third order. So essentially, from global view, they are the same. But of course, from individual view, they are different. So therefore, that means realistic. Okay. In this way, it will satisfy by theory a very nice property called exchangeability. That means, we can arbitrarily swap, this original feature and its control, right? Without undermining its underlying dependency structure. In this way, these genes and its control, they are equally likely to be correlated with noise and signal, and then they will be equally likely to be a false positive. Okay. So, and then now, once we have that, this is the global pipeline. We have this data as input, and we construct some now to mimic the dependency of these features. And then we concatenate along this feature axis to feed into this machine learning model to predict some disease of interest. Okay, so what's mentioned is that even though we double the feature by twice, but due to some very smart design we proposed previously, we only need extra overhead, computational overhead by the number of features. So let's say we have 10,000 features, and then we train a model with billions of parameters. Double the feature will only cost you 10,000 variable, 10,000 parameter. So compared to a million parameter transformer model or something equally complicated, this overhead is basically negligible. Okay. And then we train the model out. Of course, similarly, we want to do the ranking. Now we want to do ranking of both the original feature and the fake features. So as a result, we induce these interactions as a ranking list. But now if we look at the types of the interactions, there will be different type, whether they both from this original data or one of them or two of them from this fake data. So the basic idea is we want to use this fake data we already know. So as like a negative unlabeled problem to estimate what will be the false positive. This is the basic logic. So the intuition is, if a feature is really important, it should be very, rated high with high scores, and it will be way better than, the knockoff interactions. But what if an interaction, original interaction is, they are less important. They are not important at all. So essentially you will see, oh, they are indistinguishable from these knockoff interaction populations. So that will be the control, because if you want to show you are useful only if you are way better than the nulls. So this is the logic. So the selection procedure works as follows. We just look at all the pairs one by one, and we just go through, just one by one. Whenever we encounter this original interaction, this will be our denominator. This will be treated to be our discovery. Because as long as it involves a knockoff, of course, it's false. And then when we encounter this, knockoff involving interactions, either one knockoff or two knockoff, we follow this equation to calculate the estimated false discovery portion. So I skip some details to explain why we derive this equation in this form. But from high level, it follows that kind of inclusion-exclusion rules. Because we have 3x knockoff interactions compared to the original. So essentially, this is make sure that their magnitude is comparable. And now we can evaluate our method. Okay, so we start from simulation. We have a bunch of very complicated generated function, and then we generate x and y. We use this x and y to fit an arbitrary machine model and explain this machine model to guess, right? How do we identify this confidently, with FDR control, these important non-additive interactions we can show here. And then to compare to the ground truth to see if we really control the FDR at a certain level. So we can see that, the only baseline method basically is a permutation, pairwise permutation method developed by Columbia Biostat Group. They tailored for permutation, the interaction detection in deep neural network. Unfortunately, the only baseline on the market, they cannot control the FDR. And for our method, whichever method, machine learning model we use, ranging from transformer, tree model, symbolic neural network, or factorization machine, which is a very important model used in industry for recommendation system, or other models we did not show here. We can always control the FDR under certain levels. Okay, so of course, you can see that the transformer has the best power similar to recall. That means among all the ground truths, how many we can detect. Transformer has the highest power. It's not because transformer is most powerful. It's because transformer is a deep neural network model. And in deep neural network, we have some previous design layer to make sure that it achieves the most sensitivity. So although you can argue, the baseline have the highest power, but remember, our goal is, first of all, to make sure our detection has limited and bounded error. Within that condition, necessary condition, how many detection we can make. So if we fill the necessary condition, even if we have higher power as meanness, because we can simply say, we detect everything, and then with almost 100% FDR, what's the point, right? So, and we show that the reason why we can control the FDR is we use using this distilled non-additivity instead of using off-the-shelf interaction measures. If we use this interaction measures off the shelf, hopeless. Okay, we cannot. Now, next we want to apply to some real data. Okay, so this is a very well-studied Drosophila early embryo development data set to study what will be the roles like the TF plays in early Drosophila embryo development in terms of its enhanced activity. So essentially, the idea is the data will be the fold change of TFs, right? Some readout of TFs, and then the Y will be the enhanced binarized enhanced activities. We ask the question: what will be the important synergistic or repressive, like non-additive TF-TF interactions or other interactions contributing to this early Drosophila embryo enhanced activity? So of course, this is a very well studied species. So mechanistically, people know, okay, what are the synergistic interactions, what are the repressive interactions, and then of course, they are limited, and then we also use this kind of literature support. So we can see that all these the stars are people already know mechanistically they are relative, they are non-additive. Okay, mechanistically people understand that, and the reason why relatively they have higher FDR is because the sample size is relatively small. We only can sacrifice the FDR confidence. And then what I want to point out is we scrutinize two of them. Okay, so one of them called Krupil and Twist. Essentially, mechanistically, we know they are synergistic in the sense that high expression of one will make the other contribute more to the activity. So it's like a synergistic. And without looking at the literature, we simply look at this figure shows that the high expression of one will make the other contribute more to the prediction. Essentially, even without knowing any literature, based on look at this data, we know they are synergistic. So essentially, the idea is we aim to facilitate data-driven biology. That means without doing real experiment, how do we guess with a high shot, a good shot? So essentially, this provides a way to do that. And analogously, for another pair of TFs, we know mechanistically they are repressive. And just purely based on this figure, we can safely guess they are repressive, even though we don't know the literature. So in short, we provide, to summarize this part, we provide a way to detect non-additive interactions from generic machine learning models. Because this deep neural network is a universal approximator. So in principle, we can detect non-additive interactions for any genotype-phenotype mapping. Okay, so from high level, and this is very impactful. This work as well as other works. So it has been followed by many top researchers from prestigious universities, including Brown, Stanford, Princeton, and Harvard, and so on. Okay, so from high level, the method we developed enable us to provide a systematic way to prioritize hypotheses with FDR with statistical confidence. So the idea is, in the story one, we know, okay, we can do the model neuralization, model interpretation to get a hypothesis. Now we say, okay, this hypothesis can, together used with knockoffs, we can provide FDR. So, and then we can safely say, oh, these are the FDR controlled, highly confident hypothesis. Now let's do the experimental evaluation. Okay. So you may wonder, right, is this the end of the story? It turns out that it's still not the end of the story because the data we look at it's all the table, right? We have the cell by gene, cell by protein, something, cell by metabolites. So we have this underlying assumption that this data is well structured, well informative, so that we can kick off the downstream modeling and interpretation. But is that really the case? So this will motivate us to the third story. Okay, in the third story, we want to show down to the data analysis is still very, very important. Okay? And in the third story, we will do something very controversial. Okay, so we will revisit classic tools in bioinformatics. I would argue this might be the most widely used tools in bioinformatics, BLAST. Right? It compares the sequence, do the database search. So it has been proposed 30 years, with 200K accumulated citations. Many of them, they just use it, not cited. So it's undercited. And then it's used daily everywhere. So according to the New York Times, it's the Google of biological research. Okay. So as there are a lot of applications, I don't want to go through them all, because I know people here use study genomics or the evolutionary genetics, right? So there are a lot of classic use. But meanwhile, even for right now, the frontier research, people in AlphaFold, right? People try to predict the 3D structure of the proteins. BLAST is also extensively used as a data augmentation component. Say, okay, I want to find out this multiple sequence alignment so that it will augment the data to get a better prediction modeling. Okay. So this is the workflow of BLAST, in particular BLAST-P for proteins. So the query is, we have a protein, and then we have a database of proteins. We use BLAST to induce a ranking of proteins with the hope that the higher ranked proteins will be more similar to the query, right? And so by so-called alignment score. And so this alignment score, essentially, we don't need to know the exact details, but from high level, this alignment score, essentially, there will be, we try to reward the match, to penalize the mismatch or indel, with some kind of scoring scheme. This is the high level idea. We don't need to know the Blossom 6.2 or something like that. But this scoring scheme just weigh, how much we reward or penalize different, alignment, misalignment. So the reason why BLAST is so popular in the past 3 decades is because it not only gives you a score, but also give you a confidence. I will explain the definition of e-value in next slides. But basically, this e-value is something people believe statistically accurate, so that people can use that to report how significant their result is. And then maybe, publish it. So without, within the past 30 years, people just blindly use it, trust it, without have a second thought whether this is correct or not. A little spoiler alert, it's not. So let's dive into that. So from high level, what is the definition of e-value? I try to make it less mathematical. I will just explain the high level idea. So this is like, this is like if we have some kind of the alignment score, and what will be the expected number of the possible alignment with an even larger or more significant higher scores. Okay. So essentially, for this null model, which is a classical result, say that if all the query and database sequences, they are RID, and then the probability of finding alignment follow this kind of Gumbel extreme value distribution. We don't need to know the details right now, but the basic idea is, this distribution look like this. Okay, it depends on the number of, because this ID, the length of the query, the total length of database, as well as some kind of two distribution specific parameter. One is called the kappa. Essentially, it's analogous to the mean in Gaussian. Okay, and the other is called the lambda. Basically, it's like analogous to the reciprocal of the variance. So this is not important, but what really important and take away is that these two parameters, distribution specific parameter, they only depend on the scoring scheme, like how much we penalize and reward the misalignment versus alignment. They do not rely on the database or query because they are pre-calculated as a built-in parameter in the software. So they are totally independent of the database. So this makes me make us feel very suspicious because in principle, in extreme case, the query and the database, they can be extremely correlated, right? Highly similar. And will that not relevant to the estimated e-value? In principle, it should, but in theory, it's not. Okay. So which one is correct? So we did extensive simulations. In the setting one, we just choose a random protein. Of course, we did a lot, but here I just show a few. So we randomly choose a protein and then shuffled 1 million times. And because these are shuffled proteins, so they never exist in a real database. So we search against a real database, and then using some scoring scheme. And what we can see, I called it because they are equally bad. So we hope that the cumulative density function, CDF, should be uniform, right? They are equally bad. So we hope that, this CDF will be distributed along the diagonal. But what we observe is something we call the significance overestimation. That means it will overly report the false discovery they're expecting. So this is more, some numbers. For example, we expect that 5% of the alignment should have e-value less than 0.05, but essentially we detect way more than that. So you may wonder, okay, this is not a big problem because in real world, we usually use a very, very stringent cutoff threshold, say 10 to the -6. So will that solve the problem, right? At that time, we don't inflate our discovery. But unfortunately, it's not, okay? So this is another case. Still the same 1 million shuffle, but we just search another database using a different scoring scheme. Now we see something opposite. I called it significance underestimation. That means it will be way more conservative. It will miss a lot of true discoveries. A few numbers here. We want to identify 5% of alignment with e-value less than 0.05, but essentially it's only 0.8%. So it's already missing a lot of true discovery. Maybe some of that might be the driver of the cancer mutation genes, but cancer-driving mutations. If we use, let's say, an even more stringent cutoff threshold, this will risk us to lose more potential true discovery. Okay, this is bad, So essentially, in short, this either overestimating or underestimating is arbitrary. Essentially, we don't know, this is really arbitrary. To make it seem more nightmare, okay, we look at all the tools as long as it involves an e-value, okay, either different implementations of BLAST, different extensions, different settings, they're all wrong, okay. So essentially, this is terrible because it seems that we are based on a lot of findings, based on this e-value, they are based on a shaken grounding. Right? So what should we do? Okay, now we need to do some troubleshooting. We start from the beginning, say what will be the most classical result. Right? If the probability of finding this, the alignment will follow this Gumbel extremely distribution, whether this still holds. We start from the beginning. The basic classic result. The idea is, we have this 1 million result. So we try to refit a Gumbel distribution. And then to test all this, essentially we try to fit the Gumbel distribution, of all the e-values, all the alignment scores. And then we recalculate the p-values under this fitted Gumbel distribution. And what we can see is, okay, still holds, right, under this 1 million shuffle now. That means the good thing is the classical result still holds. Now, what's the problem? The problem is, in short, we are facing a dilemma. The dilemma is between the statistical accuracy and the computational feasibility. In traditional BLAST e-value, because we pre-estimate all the Gumbel parameters, it only depends on the scoring scheme. It's feasible. We calculate every day. We do this every day. But as we show, statistically, it's problematic. But if we just do, all these kind of things, fit 1 million times of this kind of Gumbel distribution, recalculate the p-value, it depends both on the scoring scheme, query, and database. It is accurate statistically, but we cannot afford that. So therefore, we propose this idea called student Gumbel p-value, which achieved both statistical accuracy and computational feasibility. And there are some extra benefit I will show later why this is more broadly useful. Okay. So the idea actually is very simple. Let's start from this observation we learned from stats 101. Okay, if we have a score, and this score, how do we test the significance under this Gaussian null distribution? Right? So if we know the parameters, say the mu and sigma, we know its parameter, then essentially we just easily standardize something we observe, and then to test the z score under this standard Gaussian distribution. But what if we don't know this parameter? Still not the end of the world. We just try to generate some null samples, and we can estimate the sample mean and sample variance. And we, instead of doing standardization, we do a similar thing, but now we call it studentization. And then we tested this t-score under this t-distribution. So it's important to take away is, so this so-called studentization applies to any families, characterized by local and scale parameter, including Gumbel. So therefore, the workflow is very simple. Okay. Just query this database, we get some kind of alignment score. And meanwhile, we just shuffle this as this is the observation. And meanwhile, we just shuffle the protein handful of times, maybe 20 times. And then we query the database to get these the highest ranked alignment scores. So this will be our null samples. Okay. So we use these null samples to calculate this sample mean and sample variance, okay, and then to do the studentization and test it under the so-called studentized Gumbel distribution. So we wrote a wrapper of the existing blast. So essentially now let's add one more column called SGP value. And then I will show later why this SGP value is accurate. So we can, one more advantage is that because previously in BLAST, it only supports a couple scoring scheme like BLAST P, Blossom 62, PAM 70, and so on. But right now, we can look at this equation. All the scoring scheme related stuff, they are canceled out in both numerator and numerator and denominator. So therefore, in principle, we can support any scoring scheme. Okay, so this is a simulation study showing that only 20, I remember, 25 shuffles, we can achieve as good as this 1 million, okay, in both significance under and overestimation scenario. And lastly, let's do a real data. So essentially, we pick a protein domain database. The task is we just try to search each sequence against itself to see what are the homologs. Okay. And then, of course, this protein domain, we have the labels, so whether this is homolog or not. And the task is, what will be the number of detected homologous species subject to some kind of common cutoff threshold, say 1%. And then we can see that in this PAM 70, this scoring scheme, which we show that it suffered this significance overestimation. That means it will tend to be too conservative. We fix that problem. So therefore, we can show we can detect more under the same cutoff threshold. And it's worth noting that another unconventional scoring scheme called PFA sum 60. Basically, this scoring scheme is try to build on the structural information. So it's basically perfectly for this domain detection, homolog detection. Of course, this is unconventional. BLAST-P or other methods, they don't support, but we can. And as this suggested, it will detect way more IDs, compared to use a conventional non-structure related scoring scheme. Okay, to sum up the last parts. So essentially, because BLAST is so important, so heavily used, so we are the first one in the past 30 years to show, this value is broken. We need to fix that, and to draw the caution that all the values are broken. Okay. And not only in BLAST, but also like right now, people do structure to structure search. They are broken. People using protein language model to do the search. They are broken. Okay, all broken. So potentially a lot of conclusion might be revisited, worth revisiting. Okay. So from high level, even though we develop a very fancy from modeling to interpretation to hypothesis generation pipeline, still a reliable data analysis is still the key. Without a good, reliable data analysis, everything downstream will be garbage in, garbage out. Okay. So in short, we aim to facilitate data-driven biology. This is very hard. The challenges, as we previously mentioned, there are three aspects. How do we trust black box machine model? We can use model neuralization and interpretable. How do we avoid overwhelming hypotheses? How do we limit us to pick too many false positives? We can use knockoff plus FDR control. And lastly, how do we deal with heterogeneous data? There's no universal rule, but each data individually was, a very scrutinized investigation. So reliable multi-omics analysis is still the key. With that, it's all my today's talk. Thanks for attention.
Maybe we could do. So I see that this is used by the trivial examples. Do you think that you could use the same method to make transportation? And if yes, how could someone ensure biology?
So the question is, how do you use the FORCE project to do differential analysis?
To do counterfactual explanation generation,
Uh-huh.
and how do we ensure biological plausibility?
I see. So right now, the counterfactual is more like something you never explore, right? It's more like extrapolation. So right now, if we look at the first story, it's more like something you already have, and then you just move around. So in order to support this kind of counterfactual generation, maybe the model itself need to be trained by conditional on something. This something is like a encoding of the different conditions, different tissues, different metadata. So therefore, it might be feasible. Yeah.
Thanks for the really nice talk. It was really shocking to hear about how like the blast over reports significance. I'm wondering if you did a little bit digging into to see if like specific types of hits tend to be more like overrepresented in significance versus maybe perhaps others were more well calibrated.
So I think the major takeaway is we cannot make this e-value only depend on scoring scheme. It should depend both on scoring scheme, query, and the database. So I hope that we can build a model to predict in a faster way, because right now we have like 20x extra cost to do the shuffles. So so essentially look at this, basically this is query dependent.
Yeah, I had a similar question with that on the blast. So in the shuffling process, is it assumed that every residue can be independently shuffled?
Yes, we just read the random permutation. In principle, it can be better. We can consider this kind of the local context to do a better, smarter shuffle.
Currently, the data may not be that, meaning evolutionarily, or the historical linkage between adjacent residues is probably much, it's not IID, but so is the inflation of the e-value because of that?
Inflation of e-value depend on, so essentially still the same, I think the e-value, the way how e-value calculated, the model is wrong. Essentially, you cannot purely rely on the scoring scheme to generate a bunch of simulations to fit this Gumbel extreme value distribution. You need to make this e-value distribution you fit, right, related to your query and database. So this is the key takeaway.
But if you only allow the shuffling pairs of adjacent things, retrievals adjacent.
Oh, you mean to fix the
possibility of that. certainly if assumption was history of lineage of the proteins.
Mhm. Okay.
I don't know if that changes.
Yeah, it was investigating, but right now I have no certain answers for that. Uh-huh. Mm. Mm.
Your model, what's the rule of feature selection? Because your embedding space actually depends on those selected features. But if you, like DC2 doesn't have this problem, because they test each gene.
Yeah, right.
So what's your like feature selection? What's the rule?
Right now, there's no feature selection. It's just a ranking.
Because for the standardized analysis, the way we do feature selection is, first is the first dimensionality. Second is because there are more noisy genes and non-conforming genes. If you don't have that feature selection, how do you show that in embedding space capture the true biology?
Oh, so it's the embedding space is, you mean for the real data? Let me see.
You need to have this cell embedding.
I see. So let me check here. So for this one, the embedding is basically just every all the genes. There's no feature selection in this figure. And then for the later on, where we show it, basically it's like a function of the number of genes you select. Because there's no, we did not provide a guidance how to choose the best cutoff threshold. So therefore, I would suggest, we can connect the first and second story together. That means we add some control as part of it and in the ranking list, and then we can cut off the ranking list by FDR. No, so the cluster result is basically we just do the clustering on the, we just use the 2 or 3. Yeah.
All right, in the interest of time, let's give speaker another round of applause. Thanks again for joining.
Thank you.